In this part, we are going to talk about a DNA damage or loss of genomic integrity brought about by a reactive oxygen species or chemicals that enhance the oxidative environment inside the cells. For example, hydrogen peroxide, hydroxyl radical, or the superoxide anion. Reactive oxygen species are generated under the influence of ionizing radiations. Unfortunately, reactive oxygen species are also generated in the mitochondria via oxidative phosphorylation. Ironical, isn't it? The very oxidative phosphorylation that generates ATP, the energy currency, leads to the formation of reactive oxygen species that induces DNA damage. Let's look at one example of mutations caused by reactive oxygen species. Under the oxidative environment, the guanosine residue gets converted into 8-oxoguanosine. And 8-oxoguanosine has the property of base pairing with A. Now, can you see the consequence of this? In the next round of DNA replication, this 8-oxo-G is going to serve as the template for incorporation of adenosine in the newly synthesized daughter strand. If this error is not taken care of, then the GC base pair that was originally located in this position is going to get converted into a TA base pair. There are other examples of altered bases generated under the influence of reactive oxygen species. For example, conversion of thymine to thymine glycol. Then we also have thymidine glycol or hydroxymethyl uracil, which is an altered form of uracil. Then we have 8-hydroxyl-2-deoxyguanosine. So what we now know is that oxidative environment leads to generation of altered bases in the DNA. But let us not forget that the cells do have a surveillance and repair system in place that is going to detect these altered nucleotides in the DNA and remove these lesions. So after removal, what happens to these altered nucleotides or bases? They are excreted and can be detected in urine. Thus, the levels of the altered bases in urine, that is their urinary output, gives us an idea about the extent of oxidative attack on the DNA. There have been prior estimates of the rate of oxidative DNA damage in humans. Going by the levels of thymine glycol and thymidine glycol in the urine, the estimated rate of oxidative DNA damage in humans is 300 hits per cell per day. Going by the estimation of hydroxymethyl uracil, the estimated rate is about 700 hits per cell per day. It means that on a daily basis, 700 uracils are being attacked in the oxidative environment in each cell. Remember that thymidine glycol, thymine glycol, and hydroxymethyl uracil are not the only modified or altered bases that are formed. There is 8-oxo-G and many others. So if you take into account a sum total of all the modified bases in the urine, the rate of oxidative DNA damage will be in the order of several thousands of hits per cell per day. What does this all indicate? It indicates that under basal conditions, there is detectable levels of oxidative DNA damage happening in our cells. In the next part, we will see how the idea of estimating the DNA damage based on the urinary output of the modified bases or nucleotides led to some very interesting results.